Welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effects Seminar Series. My name is Diego, and I'm a member of the VES Organizing Committee, along with Stephen Airwood, Mireia Selma, Jan Ilbudu, and Adrini D'Souza. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. As a reminder, we are always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I just have a few housekeeping items to mention. Each talk is about 20 to 25 minutes, and there should be time for some questions. Please post those questions at any time to the Zoom Q&A, and we'll read them out to the speaker at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we are posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can watch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. You don't have to worry about copying this URL. I'm going to post all these links to the chat once we get started. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. If you've enjoyed these seminars, then be sure to register for the Mutational Scanning Symposium, which is going to be held online and in person at the Wellcome Genome Campus in the UK on July 13th and 14th. The website listed here has all the details. Just keep in mind that abstracts are due by May 16th. For the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. Without further ado, we have an excellent set of speakers scheduled for today's presentation. Guillaume Butler-Laporte is an infectious disease clinician scientist and a Canadian Institutes for Health Research Fellow at the University of Oxford. He is a graduate of Brent Richards' Genetic Epidemiology Lab at McGill University, where he was actively involved in the genetic community's COVID-19 research response. The title of Guillaume's talk today is Slowly Unraveling the HLA, an application to vaccinology using the UK Biobank whole exome sequencing. Guillaume, go ahead and share your screen and start whenever you are ready.
Amazing. Let me share just now. That should work now. If I'm not looking directly at you, I apologize. I was a two-screen setup, but I promise trying to make this fun for you. So yes, thank you for the introduction. Really happy to be here today. This is a really interesting seminar series. So some of what I'm going to present today is on medRxiv, currently under review. A lot is actually under writing right now, and that's because I like seminar, because I don't have to worry too much about peer review, and I can show you things a bit more freestyle, a bit more looser, and just show you what I'm doing and the interesting stuff I'm doing. So I'm sorry if it's not all published. Most of it is, or will be very soon. So yes, so the game plan today, discussing HLA mutation with sequencing, and then what we did with the UK Biobank. We're going to take a little detour through the UK Biobank, then back to our problem of imputation, and then taking that last step in HLA association studies, because anyone who's actually worked on HLA knows that the big problem is that it seems sometimes that nothing translates ever to anything clinical, and it's really frustrating. And that's why I wanted to present this, even though it's not published, because it seems like maybe as a field we're moving towards this, and this is very interesting. All right, so a little background on the problem I had about 6 months ago when I started my fellowship here. I was giving a data set of a bit more than 1,000 Bangladeshi children who had received hexavalent vaccine, the regular World Health Organization vaccine. They all had the same age. They were all genotyped, and they all had their antibody measured roughly 6 months after their last vaccination. So a very nice study design. like everything was really very well controlled for. People were vaccinated at the same time, all had the same age. Really, the only major covariate was sex, really. As you can see here, the usual principal component plots, the purple and the red that you can barely see is kind of within this these South Asian ancestry 1000 genome individual. I don't show this for any other reason. It's to say that even genetically they were actually quite homogeneous. So we had maybe something we can do here to study better vaccine response. And so you knew this was coming, of course. Genetic talks. We did a genome-wide association study, and usually at this point, this is where you go, oh no, the HLA. What am I going to do about this? And usually at this point in presentations, this is where the presenter goes on a rant about 30 minutes and describing the HLA, which I swear I'm going to skip right now because I don't want to make you suffer through this. Yeah, no, let's not do that. But HLA in 30 seconds, because you still need to understand how much I struggled. Okay, this is important. I need to convey how much I struggled. The problem with the HLA, the problem, the reason why everyone hates it, or hates it, is it's got a lot of polymorphisms, and you know, with genome-wide association studies, we like the RSID system. It essentially falls apart there. There's just too many of them. People don't bother actually all annotating them. There's just too many. The other problem is that each version of each gene, and this is what we call alleles. So in the HLA system, we use the word allele not to talk about SNP, but to talk about the original Mendelian definition of allele, which is a version of a gene. So each version of the gene, each allele tends to be inherited in a very weird, specific way. And usually it's with other specific genes. A classic example, if you know about it, is DRB1, 3, 4, 5, where not everyone has the 3, 4, 5, and specific alleles for DRB1 are inherited with specific alleles for DRB3, and it gets messy really quickly. So because of that, we use alleles, not in the SNP sense, but in the whole gene version sense, to do all of our association studies and to better find map what's going on. And so the major question at this point is, we have to speak in the GWAS. How do you assign an allele for all of your HLA genes to each participant? And so one of the ways we do this is just a regular figure. You can probably see my my mouse. Can you see it? Yeah, okay, good. I just use this figure because I think at this point, most of us are kind of comfortable with the idea of imputing SNPs, right? Where you start with a reference haplotype, let's say TopMed, and you have the whole genome genotyping data with different SNPs. You know, it's exactly like that in HLA, except that instead of looking at SNPs here, you look at HLA alleles, so whole genes. So like in SNPs, you can either call an HLA allele or impute it. Let's get that one. And you've got the usual problem with imputation. So you need the reference panel. So those big set of SNPs right there. And of course, in genetics, you know, unless your cohort is European genetic ancestry, you're kind of stuck, right? So same thing for South Asians, not really well represented in publicly available HLA imputation panel. To give you an example, the biggest imputation panel is publicly available is the Michigan Imputation Server, which is a very good panel, but there's about 400 South Asians, maybe 500, something in there. We had twice that, you know, so it feels a bit strange to impute with that kind of reference panel. Of course, in parentheses, rare alleles are not well imputed, but I'm not going to spend too much time on this actually today, but just so that you know that, just like any imputation, rare alleles are not very well imputed. All right, and this is where the UK Biobank comes in. So we're like, okay, so what are we going to do at this point with this cohort of 1,000 people? Nice peek at the HLA, but they're South Asians. We don't really have a good reference panel. What are we going to do? Well, the UK Biobank, being in the UK, they have a big South Asian population. So we had about 10,000 South Asian genetic ancestry participants there. So we thought, maybe we can do something with this because, well, almost all of them. I wrote actually 455,000. This was actually the last version. Now it's up to 475, but still, we had whole exome sequences available in all of them. So we go, oh, there's an opportunity there. And so a bit of serendipity, because this is what I did in the lab. I just graduated with Brent, where we had called the HLAs for every single UKB participant of those 455,000. So when I arrived at Oxford and I was faced with those with the task of essentially finding a reference panel for those South Asians, it was, oh well, I'm in luck, or we're all in luck. I can use something from my previous lab. So that's what we had done at the in Brent's lab, which was essentially using HLA-G, which directly calls instead of impute HLA alleles. What it does in a nutshell, you take the whole exome sequencing read, you align them, you match them to different exons and the HLA alleles and to the introns, and then there's a fancy little heuristic algorithm that takes all of this and gives you an allele. Very similar to actually SNPs, really, you align and then you call, and very similar. Give you an idea of the accuracy of HLA-G in the 1,000 genome. You need to look at the column in bold. It's close to 100%. Low coverage is whole exome sequencing. High coverage is, whole genome sequencing and whole exome sequencing. Yeah, so HLA-G works really well within the ambiguity that comes with sometimes the naming convention in the HLA, but it works really well. So that's what we did. It was basically a pipeline just to give you an idea. Start from the BAM, go to the FASTA file, align the reads, weigh the reads, calls. We did that on 455,000 individual. It cost Brent about 18,000 pounds. So sorry about it. What was I saying? Oh, yes. And so in the end, we thought, well, you know, if the goal is to have a reference panel of South Asian, that is from the UK biobank, because we can't trust the ones that are out there, you know, at least let's check that our whole exome sequencing calls is better than imputed. Let's compare the whole exome sequencing calls to the imputed UK biobank HLA alleles. And what we see here, if I compare the Europeans, which you would expect would have the best concordance, which is what we see, a concordance that's pretty high. DQA1 is a problem, and I'm not gonna go too much about into this one, but DQA1 essentially in our results. South Asians, the concordance drops quite significantly, which goes with the problems we thought was happening, which was imputation panel in non-European ancestry have lower accuracy. Adjusted concordance, it's just another set of value, but essentially there's a lot of ambiguity between HLA alleles nomenclature. An allele that's going to be called in some way right now in 5 years may change because we discover another one. So I adjusted for that, and you can see that even when you adjust for that, you still see that the South Asian concordance decreases, which is, given that HLA-G is kind of 100% accurate or close to that, it really means that what we're seeing was that there's a lot of discordance in imputation between imputation alleles and called allele, and this is really problematic for non-Europeans. And so essentially we were on the right track. We were like, okay, so now we have a reference. We know that this reference most likely calls alleles better than imputation, panel imputes alleles for South Asian individual. So you can use that as our new reference. Because I just spoke about this pipeline there, I'm just going to talk 2 seconds about what we actually obtain as other results and that are on medRxiv right now about our whole exome sequencing HLA calling results. So I encourage you to have a look at it. Just a little detour because I thought, well, you know, might as well. So, few cool results. First of all, if you actually compare HLA diversity in the different genetic ancestry groups, this is based on 1,000 genome. It might not be the best way to actually describe genetic ancestries, I agree, but you know, all models are wrong. Some of them are useful, so that's why I present this. So if you actually had to guess per capita which genetic ancestry would have the most diversity at the HLA, you would think probably African, because that's what happens in, well, in the rest of the genome. It's not actually true. What we see here is per capita admixed American. Here again, admixed American, admixed American. This was to me a very surprising. We're not the first to report it. It's actually seen all over the place, and this is actually caused, we believe, this is based on the study of ancestral genomes, is actually caused by Europeans when they arrived in the Americas. The selection pressure at the HLA was greater than anywhere else in the genome. We're not entirely sure why, but well, I mean, we think we know why. It's because of introduction of different infectious disease, but we don't know which infectious disease. But I thought this was very interesting. So please have a look at my paper. If you want to see, you know, does that really increase power, whole exome sequencing? Well, this is at two-field resolution, all of the different association we could find. These are genome-wide significant. And I classified those, you know, essentially the novel ones are the dots there. And you can see that, well, we find a whole bunch, from about 340 associations, we found 129 new ones. Most of them survived conditional analysis. And as you can see, we found some that are actually, you know, quite rare alleles, you know, 10 to the minus 3. So this also goes with, we have increased power for rare alleles from sequencing. And then one last thing we did is, if you follow a bit the literature in the HLA, you see that usually they stop at two fields, so only at the exons, but they don't actually look at synonymous variants, which if you look at the rest of the genome, you would think probably matters in terms of association with different studies, and that's exactly what we see here. You don't have to look at this giant column here. It's a bit more complicated, but if you look at only this one where we had HLA alleles, we had an association with the disease at two fields, so at the exon, at the protein coding level, and then you look at synonymous variants. Well, actually, if you look at synonymous variants, some of them will cause the association to flip effect direction. Some of them, many of them actually will lead to, well, the association disappearing. So this was interesting too. I want you to have a look at the meta-archive paper. It's under review. All right, back to imputation in our cohort. Sorry about that. So yes, so now we have 10,000 participants. Well, a bit less than that, but roughly 10,000, for which we have a HLA calls from HLA-HD, whole exome sequencing, because it's already there, and whole genome genotyping. So you can actually, this is what I did for 3 months, you can actually combine all of this, fill some of the holes with imputation with TopMed, and then you have all of a sudden an HLA reference panel, which is exactly what we did. And if you want to know how well that HLA reference panel, which essentially right now is the biggest South Asian ancestry HLA reference panel, this is the next slide. So we had actually a gold standard HLA genotyping results available for about half the cohort, not for all of the genes, only those ones, okay? So the reasons why I'm not showing you more genes is because we just didn't have it. But for those genes, which are all part of the classical HLA genes, you can see we had really good concordance with our gold standard. Note that our gold standard actually did not go all the way to 3 field. There was actually even a bit of ambiguity at 2 fields. So this is actually conservative concordance, but this is a really good imputation panel. It was actually beating the Michigan imputation server for all except for DQA1, which, as I told you a bit before, was the one that was a bit more problematic. Yeah. There you go. So really quick results, what we found, and then I'm going to talk about the really interesting part with the last 5-10 minutes we have, which is translating this into something that's meaningful. So The result we found was that at DPB1 0401, those who carried it actually had increased antibody levels following vaccination for hepatitis B. We had other signal for the other vaccines, by the way, but this was the strongest one, so I'm just going to stick to this one for today. This is the usual graph that you see in HLA papers of the HLA dimer. So in blue, it's DPA1, the DPA1 protein, and green, it's DPB1, where we found our signal. The red parts of the are the amino acids residues, so the little amino acid in the proteins that are actually associated with the phenotype, which is antibody levels. And as you can see, one thing that's really reassuring is that here is the yellow is any sort of epitope that binds there. So almost all of the association we found at DPB1 was actually in that peptide binding groove where that this dimer uses to actually present the T cell and start all of this, well, not all, but a large part of the immune cascade. So that was really reassuring. We found this. We found that it matched what was happening biologically at the peptide binding groove. So now we asked, oops, what is that even supposed to mean? How can we translate this to an actual better vaccine? Because that's the goal. So the first thing we did was, does that match actually binding capacity, or if you will, or binding affinity, if you will, between this dimer and what we would expect from the hepatitis B surface antigen protein? Does that make sense biologically? Are they actually supposed to bind like we think they should, genetically speaking? So there's different algorithms in silico. We use one of them that you can use to predict epitope binding affinity to different HLA dimers. And so we use them. We took a whole bunch of hepatitis B surface antigen protein sequences, and we fed it to that algorithm. So on the right here, you actually have the result of the genetic HLA association. So that's the genetics on the right. So here, more antibodies is on the right hand side. You can see the DPB1 is the one that was the most associated. Here's the prediction. Of course, they're dimers, so it's combination with DPA1. And you can see the prediction matches exactly that, right? So what we have in silico is matches what happens in the genetics component, which is really nice. And it matches what we saw with the amino acid in the peptide binding groove. Okay, so this is very reassuring, right? Now, how can that be helpful? Well, you need to know a bit about the biology of the hepatitis B vaccine, which is that there's actually multiple different version of the protein, the different isoform of the hepatitis B surface antigen, the L, M, and S. As you can see, they're successively smaller, or shorter, I should say. The ones that are actually part of yeast recombinant vaccines for hepatitis B, and these are the ones that are used in most mass vaccination program, including the one in Bangladesh, only use the S protein. And so I'm skipping a lot of the data visualization here, but what I can tell you is if you actually feed the entire protein instead of just S inside the in silico prediction algorithm, what are you going to see? What you're going to see is that, well, this is a full protein, the red part. This is S, so meaning that those proteins that were genetically predicted to bind more, what they were actually binding to was the S part, which is exactly what the kids were receiving. And what they found less to was the rest of the proteins, but it's exactly the opposite in the other dimers. So those that were predicted genetically to bind less, well, if you gave them the full protein, you'd give them more targets to actually bind to and potentially get a better vaccine response. Now, this is all in silico. This is my last few slides. So who cares, right? Who cares? Good question. And that's the interesting part. So I'll cheat a bit. I won't talk about any animal studies or any in vitro or all of that. That probably should be done. And I'll skip straight to randomized controlled trials. Because there's this one, and this one, and this one, and this one, and this one. And yeah, that's it. But there's probably a few more. They essentially all say exactly what I found, which is if you vaccinate with a vaccine for hepatitis B that contains the pre-S, so the longer isoform, you get better vaccine response. And not only do you do that, but in this is why I put this one last, but in healthcare workers where you want 100% success rate for your vaccination and not the 5 to 10% failure rate that you see in the usual vaccine, you could actually rescue that poor response in those healthcare workers that were vaccinated with the yeast recombinant vaccine by giving them the pre-S containing vaccine. And so that I found was great, because while we couldn't really find something that was new from an RCT point of view, like essentially just replicated an RCT, essentially gives us a pathway, I think, to discover what we can do with the HLA. And so right now we're actually working for the other vaccines using this kind of approach. But yeah, I think finally we may have something we can do with the HLA. Yeah, so conclusion, other than the weird nomenclature, the HLA is really not that different than normal variants. That's what I want you to keep in mind. Sequencing versus amputation, HLA is the same problem than regular variants. Clearly, it's still helpful to impute like we just did, but just make sure you have the right reference panel, of course. Yeah, the main difference at this point is the lack of translation to real treatment, and I want you to think that we're getting there, and that because I really think we are. This is the next step, and hopefully we're going to get there. Any questions?
Thank you, Guillaume, for this excellent presentation. While I wait to see questions in the chat, I can start with a couple of my own. We have a question from AJ. AJ, you can go ahead.
Sure. That was an interesting result about the number of alleles in the different ancestry groups. I was wondering if you would maybe expect to see more alleles due to the admixture event itself. Like if it's the case that, for example, you have like 2 alleles from each sort of ancestral haplotype, would admixture trivially give you 4 if recombination happens without any kind of selective pressure argument? Yes,
100%. That is that's part of the arrival to the Americas kind of argument. So I mean, I skimmed through it really quickly, but yes, essentially what we know from ancestral genome is that they have a number of unique HLA alleles, these pre-European ancestry participants. Well, unique in the sense that you don't find them in the other, you didn't find them in the other continents. And then when Europeans arrived, well, they admixed, of course. And there was a huge selection pressure, which we presume is because of the infectious disease that they brought. But the other infectious disease that were still happening in the Americas were still there. So this is, I mean, clearly there was a mixture. There's no question. And clearly there was a big selection pressure going on. What I can tell you is that a number of those alleles are actually unique to the mixed American group. So it's not sufficient to just go by chance. But yes, like there needed to have been a mixture for this to happen so fast.
Sure. Thank you.
Another question from the chat. There should be multiple epitopes in just S region as well as the full-length protein. When you say more binding, what do you mean? Does that mean there will be more epitopes?
Yes, yeah, I realize I went fast because I only had a few minutes. Yes, so what these algorithms do, you give them the protein sequence and they chop it up in, let's say, k-mers of length 15. Inside the groove, what we know is that the epitope that binds there is usually 9 amino acid. And so you have to find a way to kind of choose the 9 amino acids. So one of the first things these these tools do is they you give them like k-mers length 15, and they find the binding core, and then they predict from this binding core and the other amino acid how much it binds there. So yes, essentially what I did on these plots is to fit a regression over all of the epitopes and how much they bound. So it's not just one epitope. There were more. I will in the paper I'm writing, there's going to be more visualization. You're going to be able to see where the epitopes were.
I can follow up with my question. So I really liked your introduction when you presented the overview between imputation versus calling of the HLA region. Because of the challenges you highlighted, would it be difficult to identify genotype using short read sequencing? Or is long read sequencing required for fully genotype at these sites?
Yeah, so it's a very good question. So there's two parts to it. One is, so the UK Biobank is coming up with the whole genome sequencing, and I'm very curious to see what's going to happen when they do this. And the big reason is, if you want to use the full HLA nomenclature system, you're going to all the way to intronic variants, which obviously I couldn't do with whole exome sequencing. And so if you really want to get rid of all the ambiguity, you would need something that also looks at the intronic side, which I didn't do. From a different project that I can't really talk to you too much about, what I can tell you is if you have a whole exome sequencing alone, there's considerable ambiguity still, even at 4 field. And we think it's specifically because of short read sequencing. And my belief, but I have nothing in terms of data to back it up, but my belief is that if we want to actually get rid of all the ambiguity and fully call an HLA allele at 4 fields, including the fronting variant, we're going to need long read. I think that's the only way.
We have another question. Have you studied gluten intolerance associations?
Yes, celiac was in there. Oops, sorry. Yes. Yeah, the reason why I had chosen these phenotypes actually is because they were known to be associated with the HLA. So I wanted to use the positive controls. You know, if I didn't find where is this one, the B57 in ankylosing spondylitis. You know, if I hadn't found that, I'd be like, yeah, I have a problem because this is supposed to be found. Yes, so celiac should be, yeah, it's there. It's one of the ones that had the most association. In the paper, you'll see there's a section about polygenic risk score using sequencing. What I can tell you is that, I mean, polygenic risk score for celiac, almost all of these of the signal comes from the HLA. It's really surprising.
Okay. So I think I will follow up with one last question. Are there plans to include your result in some larger imputation panel or try to improve imputation? Or did you do this already?
Oh,
Sorry.
Yes, so you can't do that because you can't release publicly individual calls from UK Biobank. However, we're going to do two things. One is all of the calls that were generated with HLA-HG will be given back to the UK Biobank so everyone can actually use them if they have a UK Biobank project. And two, all of the code that I use to actually take those South Asian ancestry individuals' calls and build a reference panel, that is already on GitHub. It's on a private channel, but it's already there. So you'll be able to just follow the instructions and redo it as long as you have a UK Biobank project. Yeah.
Okay. All right. Thank you, Guillaume.